really appreciate being here, and you could tell from my introduction, my early training was really in trying to increase fertility in horses, and so it's a bit of a switch to be standing here before you, but very much a part of being a reproductive physiologist is being able to stop that as well. And Dan Baker kind of told you how I got into it a little bit. It really goes back to Dr. Powers, who knew of my work with the stallions and asked me if I would do some semen testing on the elk that she was working on. And after I agreed to go in and collect semen from these elk and do a few other silly things, Dan was approached by the park as he described, and then they decided they needed somebody who would palpate wild horses. And a good friend of ours who works for BLM says there's not a living soul on earth that's dumb enough to do that, and so I volunteered. And that's how I kind of got into the work in trying to contracept the wild horses, was really starting with the very first part of that study at Teddy Roosevelt. Since then, we've been looking at several other things, and we have tried to take what was out there and all of the problems that have been discussed here today and turn them into something positive and to sort of look at it from a slightly different perspective. And this is kind of what our driving force was. PZP you've heard a lot about, know what it is, and my first introduction to PZP was to try to find out really what it's doing. As important as it is, and we know that it's blocking sperm from binding to that oocyte, my little semi-molecular brain was trying to figure out exactly which part of it was it. And we've turned out that it's actually ZP3 specifically, and that's kind of cool because that's the actual zone of protein that's on the outside of the egg, and so we know that that's what it's doing. The other drug you've also heard about, and that's Gonacon. We know that it's EPA approved as a pesticide, that it builds antibodies against GnRH, which is going to block it from having instant effect, and so we stop the mare from cycling, and so that's what makes that one work. So the pros to both of these, you've heard a lot about their ability to be administered remotely. They're both very safe in pregnant animals. We know that they don't enter into the food chain, and if administered at the right time and the right dose, they can both be highly effective. There are some cons to each one of these. The first one is with the PZP, and you've heard about the need for boosters, and something that we haven't talked about a whole lot, but it does affect their behavior, because if the mare's not getting pregnant, then she cycles on a very regular basis. Wild horses tend to get pregnant within the first 2 cycles, and they spend 11 months not cycling at all. So there's a behavioral issue there. The cons to the Gonacon are just, again, that they require a booster, and we're trying, as you heard, to figure out what the best booster is, but they tend not to cycle at all, and so they behave a lot like a pregnant mare does and not cycle on a very regular basis. So our approach then was to look at this in a slightly different way, and was to immunize against oocyte-specific factors. And the background here is maybe take you a little bit deeper into reproduction than you wanted to go, but describe what's really happening. And what's really happening here is that when the filly's born, she has all of the primordial oocytes that she's ever going to have. They don't make more. And so those are residing in the ovary. And as she reaches puberty, then it starts to draw these oocytes out of the quiescent stage, and they go through the whole process, and eventually one of these is going to ovulate. If the sperm is there, then it gets pregnant, and we have a foal on the ground. All the other little oocytes that I've got drawn on there with X's on them is what really happens. Fewer than 1% of the oocytes that the filly is born with will ever actually ovulate. And so there's something going on along the way that as a pool of primordial follicles becomes primary, the majority of those are actually eliminated naturally. And then the same thing happens from primary to preantral, and then from preantral, so on. So that's why we only end up with one. So our question was, well, what's doing this? The other thing is to look at is that the first 3 steps of this are gonadotropin independent, meaning that there's no hormones that we know of that are driving this. So by targeting a hormone, we've only got a chance of hitting one side or the other side of these. The other part is a gonadotropin-dependent system. So we looked to see what was driving this behavior and these actions, and as I said before, there's a finite number of oocytes that are contained within these primordial follicles. That means if we go in and pull them all out, then we're done. And secondly, that they're dependent on the maturation the dependency of maturation is highly regulated. sometimes by hormones, and then a lot of times by secretions of the oocyte itself. It's saying, here I am, this is what I need to have done next. So the literature kind of demonstrates, luckily, that there are two specific oocyte factors that are involved in every single one of these changes, from primordial to primary, primary to secondary, secondary to antral, and even antral to the ovulatory follicle. So it made sense to look at those and see what would happen if they were altered. So the big picture, and I probably won't go into as much detail here as I'd like to, because I'll bore you to tears, but they're all part of the TGF-beta superfamily, and so we know where they are, we know which genes they come from and why they're developed. They're all associated with cell growth and differentiation. What's really nice about this is that they are exclusively secreted by the oocyte in most species, and so it's not going to affect anything else in the body if we can target these and have their effect specifically on the oocyte. And finally, the regulation is that they are responsible for normal follicular development and potentially the ovulation rate. So the literature is pretty rich with some of the information here, and it turns out that nature kind of does this on its own, sometimes accidentally. And so we looked at some opportunities here to describe when and where they happen. And as you all know, that there's 2 copies for every gene. And so when we have a mutation, it can either be affecting only one gene or in some cases both genes. And it turns out in sheep there is a natural mutation that occurs, and if it affects only one of the 2 genes, it actually increases fertility, and those sheep have a greater chance of twinning and sometimes having triplets. If both copies of that gene are affected, then it renders the ewe infertile. There's also some natural mutations that occur in humans, and even at the heterozygote level, women are left completely infertile and they have what we described as streak ovaries. This really means that all of the primordial follicles are gone, and so that was a pretty good clue. So then we reached into the mouse, and the mouse didn't have this done naturally, but we can make knockout mice, and we can either knock out one or both copies of the genes. And if we knock out one copy, there's no effect. Mice are remarkable little creatures, and they said, fine, we don't need that much. If we knock out both of them, we decrease their fertility, but we didn't knock it out completely. But we have a really good idea here that BMP15 is in fact involved in follicular development. When we knock it out completely in several species, it doesn't affect the health of that individual, but it does affect their fertility. So it's a pretty good target. We'd looked at the same thing for GDF9, and again, in sheep, there are certain individuals who have a single gene mutation, and again, there was an increase in fertility and twinning, which means that it tended to speed up the ovulation rate or not control it, so they ovulated more than just one at a time. And again, if we knocked out both of them, we were left with follicles that completely were eliminated. In humans, there's limited research, but the 2 cases where we found identified mutations, in one case, these women were having more dizygotic twins, which means they were having 2 ovulations. Dizygotic resulted in fraternal twins, or they had an infertility, and we don't know whether these were heterozygote or homozygote changes. Again, we can treat the mouse, and we can knock this out. The heterozygotes, the mouse said, fine, don't need both copies. But if we knock them both out, we can completely leave them infertile, and there were no follicles present after the primary stage. So this really told us this is what we're after. If we can go after all these oocytes at once, then we don't have to worry about some of the issues of having high titers throughout life. Because if we can handle them all at one time, we only have to hit them once, and then we're done. And that was really the purpose of all of this. So we've looked at this in other species, and this is really how I got involved with Doug Eckery, because he'd worked with both Drs. McNatty and Jenny Jungle. And if you immunize sheep against either one of these, you'll see that they can decrease ovulation rates, but in some cases you increase ovulation rates, which means that there's a dose effect here. If we give them enough, we can stop it. If we don't give them quite enough, just like in the heterozygote, we actually allowed things to speed up, and they had multiple ovulations. In any case, we ended up with abnormal ovaries. And so we knew, again, that we're simply targeting that, and maybe the best news here is that we're targeting the ovary, potentially the oocyte, but we're not affecting the overall health of the individuals. When this was investigated in cattle, we saw that there were no differences in ovulation rate, but a decrease in the size and the number of follicles. And so there's an argument there that maybe if the follicle still ovulates, the health of the oocyte might be altered, or if there's not sufficient corpora lutea, then there's not enough progesterone anyway, we're going to affect fertility. If we knocked out BMP or treated them against BMP, we increased or decreased ovulation rates again, telling us that there's definitely a dose effect here, that we've got to have a high enough level to stop it altogether, and just a small amount, the body probably tries to overcompensate, and they have multiple ovulations. But again, there was a decrease in the size and the number of antral follicles. This was taken into wildlife, and looking at this in deer, found that if deer were treated to be immunized against GDF9, there were no differences in fertility rates in the first year, but they became infertile after the second and third year. That really piqued our interest, right? That's not at all different than what we've just talked about. When you treat a pregnant animal, don't see anything the first time, but after that, we're affecting things. BMP15 increased fertility, and there was a higher incidence of them having 2 and 3 offspring at a time throughout life. And so we knew we were affecting, or there was a potential here to affect oocyte development, follicular maturation, and or ovulation. So these two things make some pretty good targets. So we wrote this up, and we decided that we might try to immunize mares against BMP15 and or GDF9. Our hypothesis was that vaccination against these would prevent ovulation and or accelerate the depletion. So the idea here was maybe we were going to put mares into a premature, what we call menopause, and get rid of all their oocytes to begin with. And if we could do that with the single dose early on, then we don't have to keep up titer for life. We wouldn't have to recapture them and do anything like that. And if they're not developing follicles, they're not cycling, and so they wouldn't have the same cyclic effect that we have if they were just simply not pregnant. So these were the materials and methods for this series of studies. On the third year, but for the first 2 years, we had 30 horses, and so I was glad to hear what we just learned about having to have 10 horses in each group. We had 10 horses in the GDF9 group, 10 in the BMP15 group, and 10 in the control group. We vaccinated them 4 times. Again, was a proof of concept here. We've got an adjuvant we know that will keep the titer levels this high for that 18-week period, but we wanted to do them slowly so that we didn't have potential effects of overstimulating them just in an immunological sense. We tested antibody responses every 2 weeks throughout this entire period. We also followed follicular activity by transrectal ultrasonography 3 times a week, and we followed their behavior by teasing these mares to a stallion 3 days a week as well. To make the vaccine, what we did was we used peptide regions of both BMP15 and GDF9, which were used in the studies that I talked about before in the sheep and the cattle and the deer. They were attached to KLH, which has the blue protein that you've heard about in it. Then we used PET-GL-A as the adjuvant because we didn't want it to be a really strong adjuvant. We wanted to have a mild response. The control vaccines got really the same thing again, but without the peptides. The peptides are listed here. We had these synthesized, so they're 100% sure of exactly what it was that we were treating these mares with. We based these on the known sequence of both of these proteins in horses, so it was 100% in the horse, 80% to what was used in the sheep and deer for the BMP15, and again, it was 100% homologous to the horse because that's why we designed it. And in that case, it turned out it was also the one that was 100% homologous to those that were used in the sheep and the deer studies. So this is kind of what our setup looks like. Over on the side there, you can see we call our Tease Rail. The mares will run in at least 3 times a week, walked a stallion by, and he teased each and every one of these mares individually. And then after that, the mares were taken into the palpation stocks, and we ultrasounded these mares, and these were the outcomes we were looking for. We wanted to see if they had normal follicular growth. Each and every follicle was measured and recorded. Wanted to know if those follicles ovulated. We could tell that by the formation of the corpus lutea. That was also measured. Or if we had any abnormalities in this whole process, the hemorrhagic follicle, which is the one in the center, you can see the echogenicity in there, so there's some fibrin in there, and these may or may not ovulate. An anovulatory follicle, that one's one that we've tracked for 2 or 3 weeks at a time. It got really large, got small, but it never luteinized. And in some cases, we had a hemorrhagic corpus luteum, which we would expect for about the first 12 hours, but these maintain that morphology for out to 2 and 3 weeks at a time. So the results here were to first see what the antibody responses were like. The bottom lines in both of these were the control mares. We had a kind of a strange thing with our assay. You'll notice after the second dose, all of our horses had an increase in both BMP15 and GDF9 response, but never above the level that we were looking for. And in every single one of these vaccinations, you can see a typical immune response. It peaked at about 2 weeks, started to drop off, peaked at 2 weeks, dropped off, and did that each and every time. So we know that we were building immunity, that we had the right antibodies in there for both of these particular vaccines. So this is the real exciting part about this. The control mares are here in the middle on these next couple of slides, and you'll notice that on average the control mares had 10 CLs, or they ovulated 10 times in this first year. The BMP15 mares ovulated 5 times. They were significantly different. So we've decreased fertility arguably by 50%. The GDF9 mares didn't have the same response. Their ovulation rate was absolutely identical to that of the control mares. Also, like we'd seen in the cattle, anyway, where we were able to track the size of the follicles, the average size of the follicle the day, 2 days before they ovulated was 35 millimeters. And that's been reported in the literature over and over again, so that matched quite nicely. And in both cases, the size of the follicle prior to the ovulation was decreased. And so we are affecting, particularly in the BMP15 mares, not only are they ovulating less often, but those follicles that are ovulating are smaller. So we're clearly hitting the target. This shows each and every mare on the study. The B stands for the BMP15, the C are the control, and the G are the GDF9. And then we just listed them A through J on each one to protect their identity. Thank you. So you'll notice that in the BMP15 mares, we had 2 mares for that entire first year that never ovulated at all, and 2 that only ovulated once, and of one of those, it was an abnormal ovulation. So the white are the abnormal or the smaller ovulations, and the other ones were normal. So we're having a major effect in the BMP15 mares. The kind of a normal distribution in the control, a few that were abnormal, and then you can see that they really don't differ much from the GDF9. Okay. This is to show that not only did they have smaller follicles, but the CLs that resulted from those, again, were abnormal in both the treatments, whether it was GDF9 or BMP15. So we're pretty excited about this to begin with, because what we were hoping to do was exactly that. The mares were healthy, had no problem with these horses. Matter of fact, they're still doing quite nicely. We were able to affect just ovarian function in the first year. Okay, so the results from that year, the Estrus Cycle, the behavior was not different in the GDF9, which doesn't surprise us because they ovulated just as often and the follicles were fairly similar. The BMP15 mares, we had fewer days observed in estrus, and so we changed their ovulation pattern and their behavior matched that, which is exactly what we were hoping to see. Okay, so what happens in year 2? Well, I don't know. Talked about that already. So we published that, and now we're looking at the data for the second year. Again, same mares, and we just followed them for year 2. They were the same control, GDF9, BMP15, and the data I'm going to show you are really only 5 from each group, because something that we haven't talked about, because we haven't done it yet, was we unilaterally overectomized half of the mares after the first year so that we could do follicular counts and see if there were a fewer number of primordial follicles left in there after having done that. So the 5 mares in each group that were left intact, we followed through, again, behavior, ovulations, follicular dynamics, the antibody titers, and we're also working on steroid hormone production to make sure that that matched what we saw with the ultrasound. Pretty exciting data here as well. Control mares ovulated over the 2 years, average of 20 times, and of those abnormal Ovulation's pretty similar. The BMP15, again, we've decreased the overall number of ovulations, and it turns out after the second year of the GDF9, the ovulation numbers weren't different, but there was an increase in the number of abnormal ones. And so, again, we're pretty excited about what we had here. Take that one step further, and then we followed how big these follicles were, just as we've done in the first year, and just like we saw before. Both treatments had smaller follicles at ovulation, potentially telling us that we're affecting that whole process, and they may have not resulted in fertile ovulations. That we just simply don't know. Okay? So we're now on the third year, and we've pretty much eliminated the use of the 30 mares we had from the two of those, and we decided to combine the BMP15 and the GDF9 together in a single vaccine using exactly what we'd done before. We just combined both of those peptides with the KLH and the PET gel. We treated 10 mares again. We first treated them in December 2017 and then gave them the 3 boosters as we talked about before. They followed the exact same protocol we discussed with ultrasound, demonstrated estrus, hormone levels and antibody titers. This one shows you what the antibody titers are. You can see that they all reacted as we expected. We got a boost each and every time, and we were able to maintain them out at a fairly high titer level and what our target was. Okay, so the behavior for the control mares is like we expected. It was fairly regular once the breeding season started about mid-March in Colorado. We had multiple events. They tend to show estrus about every 21 days, and each time they showed estrus, this was associated with an ovulation. And every single one of the 10 control mares showed all, we put them on a scale of 0 to 5. And 0 was a mare that was ready to kill the stallion, do whatever she could to make sure that he knew that she wasn't interested. And then a scale 5 was a mare that was showing every indication that if she could just get out of this pen, they would have a good time. Okay, and this is kind of how we charted it, 0 to 5, and you can see the dates. And these are just a couple of the control mares to show you. Very regular, very typical in heat, out of heat, in heat, out of heat, exactly what we'd expect to see. When we treated the mares with the combined vaccine, their behaviors were quite sporadic and erratic. There were multiple events in many mares, and none of the events that we talked about were actually associated with ovulations. And so, don't know if these were learned behaviors. I should have said all the mares that we used were aged mares. They were between 8 and 15, and they had all had a foal within the last 2 years. They came out of a embryo transfer recipient herd, so we knew their history for the last 5 years. Only one mare in the 10 that were treated actually showed all 5 signs of estrus during this entire year. So that's what they typically look like, showed some signs, then nothing, and then maybe a few more, then nothing. This was the one mare that on one day in, I think that's August, showed all 5 signs of being in heat, and it was only for a single day. Okay. So the next one is really what we're after. Did they ovulate or didn't they ovulate? This is what we saw with the control mares. All 10 of these mares averaged about 10 ovulations per year, like we'd seen in the first 2 years, and not a single one of our double vaccinated mares have ovulated yet. So we've got a full year of data without a single vaccine. So we're pretty excited about that. There's a big asterisk there because that's about as statistically significant as we can get. Our hopes are to carry this out for another year or so to see exactly what's going to happen with these mares. Our intentions are not to treat them again and just see how long this complete acyclicity lasts. We're also hoping to do some fertility trials. We've got a grant in right now to treat these mares one time with the adjuvant that we talked about, turn them loose with some stallions, and actually prove that if you don't ovulate, you can't get pregnant. And find out how the herd dynamics work, and then eventually the dose that we were using is about 1.5 ml or 2. This could potentially have some possibilities for remote delivery as well. And I'd also like to finish here with some thanks, obviously, to the Bureau of Land Management. If I didn't state that to begin with, this was a grant from BLM that funded all of this. Several graduate students have worked on this, technical support, and then finally Abraham Equine, which is the group that provided us with these horses and allowed us to take his recipient mares and basically render them infertile, which aren't really useful to him at this point now. Thanks.